Our next session is on the topic biotechnology to drive process efficiencies and co-product valorization for grain ethanol plants. And please allow me to introduce to you our next speaker, Dr. Ben Fuchs. He's here with us. Let's give him a huge round of applause as he joins us on stage along with his team. I would also request his team to be present on the stage with us. Dr. Ben is the Strategic Portfolio Director for Grain Processing at IFF, with over 20 years of experience in industrial biotechnology and process development. He holds a Ph.D. in chemical engineering from the University of Karlsruhe. Is that pronounced correctly? Thank you. And an MBA from Drexel University. He joined DuPont in 2004 as an expert solid liquid separation and particle technology, and led many technologies and process development. Later, he had KRL the Butamax biobutanol facility as general and technical manager. A very, very warm welcome to you and to your team. Ladies and gentlemen, a hand for the next session led by Dr. Ben Fuchs.
Can you hear me? Okay. Well, good afternoon, everyone. I hope everybody enjoyed their lunch, and thank you very much for the kind introduction. I would also like to introduce the IFF team here on the stage. First, Ritu Bala. She's the Regional Application Development Leader here in the region, and we have Arun Kumar as the Sales Leader for the region. We made a deal. I present, they answer your questions. Before I get into the topic of biotechnology to drive process efficiencies and co-product valorization in grain ethanol plants, which I think is a very timely topic considering the speeches yesterday, the speeches today. A lot of it is about process efficiencies. A lot of it is about co-product valorization. I did want to thank Jima for organizing an amazing event, connecting the industry, but also globally, and pulling in the right stakeholders so that we can have meaningful conversations which hopefully help drive the industry forward. It's also a privilege to be here and have a slot to present, which really gave me the opportunity for the last week to travel the country, visit with customers and also partners. And while I understand there are challenges in the industry, I think it's an amazing and exciting time with genetically modified yeast being trialed, with corn oil recovery technologies being implemented. I think there will be a lot of growth, and hopefully we can bring global learnings into the industry. I have 4 parts to the presentation. First, a brief introduction about IFF and the business. Keep that very short. I thought I'd give a little bit of an overview of corn ethanol in the US, which is really a story of continued innovation and optimization. We have seen some metrics related to this already. Then move on to biotechnology in ethanol production, specifically the role and power of enzymes and yeast for fermentation, for corn oil recovery, for co-product value increase. And then I close with a summary. Couple of words around IFF and our business. Our purpose is really making joy through heart, creativity, and most importantly, science. We have R&D facilities across the globe. We're reinvesting a lot of our revenue back into R&D, and we have local application development centers all across the globe to make sure that we're very close to our customers and bring the latest technology, which is fit for purpose, into the region. Our commitment is to do more good and making a difference for people and planet. Under this tagline, you see a lot of different categories. I want to just focus on the sustainable solutions. So really focusing on innovations that transform industries and empower our customers to reach their sustainability goals. And I don't know that any industry but the biofuels industry is representing this better. So I'm very proud to work in that industry and drive sustainability goals forward. Some people in the audience maybe might have thought in the past, what is IFF? Who is IFF? I will not go through all the details, but I think the key point is if you hear IFF, it is synonymous with DuPont Biotechnology, with Danisco, with Gen&Core. So those technologies over time have come together and are now residing in IFF. There are a lot of milestones here, but just a couple to highlight that we have been pioneers in the industrial biotech space. Gen&Core was formed about 43 years ago. Shortly thereafter, the first patent for protein engineering enzymes, and shortly thereafter, launching protein-engineered enzymes at large and economical scale. And throughout the timeline, you see a little bit more how those companies came together. But again, if you hear IFF, think about DuPont Biotechnology, think about Gen&Core. Now, within that company, we have a business called grain processing, which we are representing here. And what we do is really help our customers efficiently and sustainably turn renewable raw materials into solutions which matter. And we do this through enzymes, but also through yeast. And depending on the region, maybe depending on the time of year, different feedstocks are relevant. Not all inclusive, but certainly barley, wheat, cassava, rice. You all have mastered the rice ethanol. And also corn, which is growing here in India. But beyond just thinking about grain processing, think of us as a company and somebody who has a toolbox which can be applied across the agricultural supply chain. We have a large enzyme portfolio which helps us to degrade or modify the components in the agricultural feedstock. This could be starch, protein, sugars, fiber, and so on. We combine this with a broad level of and deep know-how around the substrates, around the end product features, may it be in animal nutrition, pet food, food and beverages, and also processing knowledge, agricultural processing, fermentation processes, of course. We produce our own materials through fermentation, but also support customers, and food ingredient production. And then you combine this with an innovation machine, which is not only focused on the solution development through application development and the supporting R&D, but also tries to combine it with process technology. And when you think about optimization of certain things, including corn oil recovery, I think the joint approach of biotech and process, plus some others, as I will show, play a significant role. And then we get to the commercialization. You can do a lot in small scale. Ultimately, you need to bring it to large scale, different capabilities and functions needed to do this. And just one more word around the end product features. I mentioned that grain processing is one business in IFF. We have food biosciences and animal health and nutrition as well, which have access to customers, access to knowledge, which is relevant also for the ethanol industry, where you can produce feed products as byproducts, and who knows in the future, certainly in the US, some facilities also food. Our innovation approach here for enzymes is, let's say, twofold. We obviously try to start with the customer need. That's why we have representation in all the meaningful regions to stay close to the customer. We bring those needs back into the innovation machinery and try to identify if we have something which can serve that need or meet that challenge through a data-driven approach. And if we have something, then we go the bottom path here. We're working on existing enzymes, existing molecules, and maybe we blend them so they meet your specific targets, your specific needs of your facility in your location. We then go through application development, scale up, work closely with our customers, and also support the ongoing optimization. Now, if we don't have a ready-made solution, we go through the top path, which is really the R&D path. We focus on discovery of new enzymes. Hopefully, we find something in nature we can use. Very often, it's just not good enough, where we then start with protein engineering to make the enzyme better until we have a molecule which meets the needs of the customer. But that is really only half of the story. The other half of the story is really to increase the scale of this and produce the enzymes at commercial scale. So let me now go into a little bit of information from the corn ethanol industry in the US. And again, it's all about a story of innovation and pushing the limits. And you have seen some of those metrics before. So on the left-hand side, you see the historical denatured ethanol yields over time, where you see a continuous increase. Sometimes you see a little bit of a dip depending on economic conditions, how the plants need to run. If I was plotting not the average, but the top performers, you would have seen a continuous increase. That number would say you're at about 92% of what's theoretical possible in those plants when it comes to ethanol yield. The other important metric, and we have seen this, is the historic distillers corn oil yield, where you also see the same trend, increasing yields over the years with learning more about corn oil, with new technologies coming online. And we're now here as an average at about 55% of theoretical yield. That is the average. They're high performers, as we have seen earlier. They're low performers. Also, what I want to say is that these numbers come from an internal benchmarking study we do in the United States. So I think one takeaway is 92% of what's theoretical possible on ethanol, 55% rough numbers, what's theoretically possible for corn oil. So a lot to do on corn oil and a big opportunity. And I think it was good that it was already discussed how corn oil might impact DDGS price or doesn't. And I think the US industry has become a lot more relaxed about this because of the value of corn oil. So I referred to a benchmarking study, which we do every year with our customers in the US, to just give you a flavor. This benchmarking study is going on for 10 years already. We have been working throughout that with about 127 plants where we get data from, 51 metrics we're focusing on. They change over the years, and 90 analysis we do with that data. And it's a really powerful study because we understand where the industry is going. We bring some of this information back to drive innovation, but in a confidential manner, we can also tell our customers how they're performing. How do they perform on yield? How do they perform on corn oil yield? How do they perform on energy? How do they perform on spend? And does your spend correlate with actually performance? And that would be spend on, let's say, the biotechnology. So very powerful study for us. I give you a couple snapshots of that study. What you see here is the ethanol yield and notable correlations. And I just highlight one, which is the glycerol. So as you see here, again, this is from multiple plants across the US. As glycerol increases, ethanol yield decreases. It's common sense, right? It's carbon, which needs to go one way or the other. The question maybe is more how, if I'm at high glycerol, how can I get to low glycerol? If you have the flexibility, choice of yeast can be one way, or fermentation optimization can be another one to change a little bit the environment and maybe have less stress on the yeast. Another correlation here is more for corn oil. And what you see is basically the average corn oil recovery for plants which are not using cellulosic enzymes and plants which are using fiber loosening enzymes. And you see clear improvement, probably about 10–12% between those plants. What is also interesting for us and can be leveraged in a way, or at least then can help predict where the industry might go, is really how is technology adopted by those customers. And one example I want to show here is the adoption rate of thermostable protease enzymes in the ethanol industry in the US. Thermostable proteases are used to basically generate nutrients for the yeast in fermentation. They can reduce the urea requirement, for example. I think yesterday we heard about urea contamination, I guess, in DDGS. So you can reduce urea, maybe drive it to elimination, but you can also improve corn oil, and I get into this in a little bit more detail. But what you see is that over maybe 5 years, the industry went from 60% adoption to 90% adoption of thermostable proteases. So if I just summarize the US corn ethanol production in a couple of key themes maybe. The first one is that continued drive to higher ethanol and corn oil yield will remain. New technologies are emerging to support this drive. I think you see the same with corn oil. So what we see in the US as a couple example is corn oil, the machines, centrifuges, which are used for corn oil recovery, and where the corn oil is recovered in the ethanol process is changing, and there are multiple options. One thing I would say, if you combine process with enzymes, the enzymes we add to the system in fermentation, so upstream of corn oil recovery, are agnostic to whatever separation technology you use. If we achieve more free oil, I'm sure the separation companies will be able to recover it. But what we also see in the US is basically fractionation technologies, fiber removal up front before fermentation, fiber downstream. Whenever you remove something, there is the chance you remove more than you want. Fiber goes out with a little bit of starch, with a little bit of fat. That's again where our toolbox, where we can separate those different components in agricultural feedstock, can play a substantial role. And I think I saw a similar chart a couple times maybe already today. The last thing is really a holistic approach required to maximize the value for ethanol producers. And how I describe it here, it was a little bit different earlier, is biotechnology, process, and chemistry. I guess if you take corn oil, it's enzymes, separation technology, and de-emulsifier. And I think the power of working closely together is to really get the last little bit of ethanol / corn oil out of the process. And biotechnology certainly plays a significant role. So let me now switch gears to biotechnology in ethanol production. A couple themes here. It's all about the role of enzymes and yeast. First, a little bit of background around enzymes. I promise I will keep it short because I think many of you know it. Then go into more detail for two main enzyme categories: starch-degrading enzymes and fiber-loosening enzymes. Then I will give you an overview of some other auxiliary enzymes, which are heavily used in North America or the US already and in other places, and probably will gain importance here over time. Then a couple slides on yeast, but we will have a longer yeast talk later. And then I will close with a summary after that. So here is a high-level flow sheet of a typical ethanol process, which you are obviously all familiar with: liquefaction and simultaneous saccharification and fermentation. To just highlight where enzymes are mainly used today in the process. Everybody is familiar with alpha amylases and glucoamylases used in liquefaction and SSF. You can't really run your process without your enzymes. So these are processing aids which are required to make the process work. But then there are other enzymes, auxiliary enzymes, proteases, phytases, and cellulases, and I want to introduce them a little bit more to you today. And then obviously you have the yeast in fermentation. So a little bit of background around enzymes, and again, many of you or all of you are familiar with this, but enzymes are proteins made from amino acids. They're produced from living organisms, but they're not living, and they're biocatalysts to speed up reactions. They're used to overcome an activation energy. But why do you use enzyme? They're highly specific to a very specific reaction, and the reaction can take place under mild conditions, which can be beneficial. When you think about protein can denature, milder conditions beneficial. They have very high reaction rates and they don't change or they're not consumed over the reaction. The schematic here is very simple, right? You have the enzyme in dark blue. It is a dynamic and flexible structure. The substrate docks in, the enzyme maybe adapts a little bit to the structure. The enzyme does its magic, which is catalyzing the reaction, and it basically pushes the products back into the substrate. Sounds very simple, but when you think about the use of enzymes, there are a couple considerations. There's certainly the reaction time you're trying to pursue. Maybe you have a limitation in your process you need to meet. Activators, inhibitors, substrate concentration to consider, and the enzyme activity and dosage, so you understand how much do you need to add to basically get to a desired outcome. But two, I want to highlight a little bit more because they also play a big role, obviously, in fermentation and in ethanol production, is pH. What you see on the top graph here is the relative activity of an enzyme. In this example, the enzyme has the maximum activity at a pH of maybe 5.5. And if you go right or left to this, you lose activity until the enzyme is not active at all. The same thing is true for temperature. In this case, that enzyme has the maximum activity at about 50 degrees C. And you go right or left from this temperature, you lose activity until the enzyme is dead. There's also another temperature aspect to consider, which is temperature stability. So if the enzyme is exposed to temperature in the reaction, does it lose activity? What you see here is that at 40 degrees C, it barely loses any activity over 300 minutes, but at 50 degrees, it loses some activity. All of those aspects need to be considered when you think about using an enzyme in your process. So let's now go a little bit into different enzyme classes, starting with starch-degrading enzymes. You see a picture of a starch molecule or illustration of a starch molecule. Let's start with alpha amylases added to liquefaction. You can break down the starch into smaller molecules, dextrins. There are debranching GAs and polyanases, which preferably break the molecule at the branching point. And then obviously you have glucoamylase, which makes glucose, which then can be fermented and consumed in fermentation. There are also fungal amylases and beta amylases, which can get to a 2-glucose molecule maltose. One thing, though, to mention is I highlighted some of the individual enzymes, but very often in the industry, when we say glucoamylase, add glucoamylase to SSF, very often that is a blend of different enzymes, which includes fungal alpha amylase and maybe some auxiliary enzymes. So it's more than just glucoamylase. Highlighting some of our developments over the years for one of the starch-degrading enzymes, which is alpha amylase. You see a multi-generational development. Spezyme Pro here on the market, HN and other alpha amylases coming into the market. But really it was all about focusing on the need of the industry, which was viscosity reduction in liquefaction, maybe allowing you to go to higher solids. Starch solubilization to help the ethanol yield, thermostability to be a little bit more robust, and also lower pH and lower ions over time. And that development will continue for us, and we put significant resources into the multi-generational development of these types of enzymes and others. But even if you have a lot of really good starch-degrading enzymes, there is always a little bit of starch left. So here is the starch composition in several DDGS samples, just from a literature resource. The point really is that some starch cannot be hydrolyzed during liquefaction, and some starch is bound to fiber, inaccessible to starch-degrading enzymes. So then the question is really how to get to some of this, and one way of getting to some of this is to work with enzymes to loosen the corn fiber structure. Corn fiber is a combination of cellulose, hemicellulose, a little bit of lignin. And if you go a little bit into more detail, then you see on the cellulose side, you have glucose molecules which are connected, and you need certain enzymes to basically break that structure up. On the arabinosilane, you have arabinose and other molecules, xylose connected, which you have to break up with a different set of enzymes. And earlier, I think there was a reference to Gen 1.5, and I agree. When you think about Gen 1.5, you really have to be clear about the definition. But one question could be, how do you get to those glucose molecules in the regular 1G ethanol process? So then, what benefits do these enzymes which loosen corn fiber really bring? They can bring multiple benefits. The first one, increase in ethanol yield. You get to the starch. Maybe you get to the cellulose. Maybe you get to Gen 1.5. But it can help to get to the starch. You can get to the cornol, and you can improve the mechanical dewatering and reduce viscosity. And I will highlight a couple of them. First one is cornol. So the path to increase cornol, this is mainly from a biotechnology perspective, is to understand where the cornol sits, which is packaged in tight little spherical droplets called oleosomes, protected by a protein structure and layer. That's where proteases come into place to free up that cornol. And the oleosomes are small droplets, very difficult to separate until you maybe join them together. But they're also embedded in a cellulose, hemicellulose structure. That's where the fiber loosening enzymes come into play. You all know this by now, right? A bottle which you centrifuge in the lab, there are different fractions of oil. There is the free oil at the top, easy to recover with a centrifuge. There is the oil associated with the suspended solids. Well, they're with suspended solids, so you probably have to disassociate them if you wanted to access it. There's unrecoverable oil in the aqueous phase in the emulsion. Maybe you can break the emulsion to get to the oil. And there is oil which is in the cake, which is basically lost in the whole stillage centrifuge. And when we saw a similar diagram earlier, when you then think about what matters, how do you get the corn oil out? We certainly did a lot of fundamental studies to figure out what impacts corn oil, and these are just a couple of them. Density differences we talked today about. Droplet size matters in centrifugation, Stokes' law, as you heard. Surfactants, emulsifiers, concentrations, which keep them in emulsion. External forces and temperature. And what I want to mention here is that very often we focus on the temperature, on some of the external forces in the moment, but what also can matter is the history the corn oil, for example, saw throughout the process. Viscosity, salts, and electrolytes are a couple of the other examples. So many factors, probably more than I showed here, and understanding them, fundamentally studying them, helps us optimize the process, but also helps us optimize the research, which actually goes into enzymes. And here you see an example where we went with a corn oil yield of a baseline and introduced our generation 1, which is commercially available enzyme, increased significantly the corn oil yield, and then we're pre-commercially trialing in the US another enzyme which gets you to about 30% above baseline. These are large-scale tests. But also exciting to us, a little bit of a scientist from history, is that we are also working on a third generation. And you see the baseline here, and you see the improvement in corn oil is the length of the bar, basically. Every single bar is an enzyme, and every color is an enzyme class. So maybe in the future, we're not only talking about proteases, cellulases, silonases. So let me then quickly switch to the dewatering benefits you can get. When you use these fiber loosening enzymes, you can reduce the mass of the cake which comes out of the whole stillage centrifuge, or the water which comes out of the whole stillage centrifuge. And the result of this obviously is you push more into thin stillage. So this is how you can impact the dewatering, the mechanical dewatering. But then you probably ask yourself, well, I understand what it does to mechanical dewatering, but what does it to the overall plant? So we have significantly invested in digital resources, and for the US, where it made a lot of sense, have predictive models for the entire ethanol plant. And we're using those models with the input of what we want to know. So we solubilize fiber to increase the centrate dissolved solids. We then have a centrifuge which performs better. We put this into our model. We see exactly how the process, the mass balance changes in the process, and also what impact it has on energy. In this case, 77% reduction in natural gas. That's a metric for the US, right? You use other energy sources, but it gives you directionally what these tools can do to really see the impact holistically. Then we can also impact viscosity. What happened here is that we used enzymes which allowed us to increase the solids in the evaporator and the syrup by 5% without impacting the viscosity, which can be big for a plant. The same thing we did here. We used our model to basically understand what that does for the plant. Again, cellulases to reduce viscosity, increase in syrup solids from 32 to 35% in the evaporator. We held the corn feed constant. You see all the impact it has directionally on the individual unit operations, and ultimately a 1.5% natural gas reduction. Earlier, it was also mentioned that we do a business in Europe as well. Energy costs significantly higher in Europe. And what we can also do then is translate all of this into a difference in carbon intensity score. On top of these two enzyme classes, starch-degrading fiber loosening, you see other auxiliary enzymes. We have phytases, which hydrolyze phytic acid, which is a strong chelator. And when you use it in dry grind, in corn ethanol plants, you can basically enhance the pH in liquefaction and the stability of your alpha amylases. Accordingly, we have blends of those two enzymes. We can maximize the level of free phosphate in the residual DDGS, which can be beneficial, and reduce fouling throughout the process, for example, evaporator train. Proteases, they hydrolyze protein matrices in the corn kernel that bind the various fractions. I mentioned a lot of this already, so you can get to the hard to hydrolyze starch, you can free up cornol, and you can increase the levels of nutrients the yeast needs to perform in fermentation and potentially reduce your chemical consumption, urea, for example. And similar to, so that was about enzymes. Now switching gears, just a couple slides about yeast, but we will hear a lot more about yeast in the next presentation. What is yeast, right? It is a Saccharomyces cerevisiae, most commonly used species of yeast for commercial fuel ethanol production. It is a very strong fermenter, can go up to very high concentrations of ethanol and just produces when there is sugar available. It is fairly robust already. It's in its non-genetically modified form, and it has some simple nutrient requirements. Over many years, we obviously have focused on also genetically modified yeast in IFF. You see here kind of the journey of multi-generational genetically modified yeasts we have developed. And we have developed them with multiple tools, which are important. For example, breeding of the yeast. For example, genetic pathways, which are internalized into the yeast to basically make more ethanol out of every little glucose the yeast consumes. Sunarshad Jade is in the country. We're trialing soon, so a very exciting time to see genetically modified yeast for the plants who can use it really penetrate the market. And hopefully we see a quick adoption, because ultimately the benefits are real. Climbing this journey basically enabled us to increase the yield by 4.5% over non-genetically modified yeast at the time. Now, similar to enzymes and the holistic process model, yeast is not a standalone biotechnology tool. You need enzymes to make the yeast work. So how can you basically use tools and digital tools to predict what is the best combination for customers? So what we have done here with the information we generate in pilot scale fermentations, we have developed the technical information and the kinetics and equations which go into mathematical models, which then help us how our yeast with our enzyme combination behaves in your plant in the US. Here you see an example of our tools, just some screenshots, but now you have these mathematical models or digital twins or kind of sophisticated video games, and they can answer real-world questions. So which yeast, which enzyme combination should you use in your plant? And this is all housed under our ExcelisAI digital platform. And what you see on the right-hand side in the lines are the model predictions versus commercial data points. So very predictable or predictive, and it can help us to accelerate trial planning, to accelerate optimization at the plant, but it also helps us feed back your conditions into our innovation cycles. And with that, I will close with one more slide just in time, and that is the summary. Our continued commitment to the ethanol industry in India. First of all, we provide the right biotechnology which is tailored to the specific needs of the region and customers. We have a big innovation pipeline, we have a big product portfolio, we have a systematic approach to basically bring that portfolio to where it makes sense. We leverage the extensive learnings from the US to the growing market. You have seen our benchmarking study and obviously many people out in the field. We work very closely with our customers and partners to understand the specific circumstances to ensure maximum chance of success. And I think on the corn oil presentation earlier, you have seen some methodologies. We also work very close with the customer to really understand where the corn oil is going before we bring in enzymes to see if we can direct the corn oil into the right direction first and then free it up when it matters most before its separation. We provide technical service in the field to ensure that these maybe benefits you see from a first implementation of biotech will continue and are sustainable. And we're committed to innovation to further increase the value generated from each kernel of corn. And what matters here too is the entire co-product valorization, where a lot of efforts are going on in the US, and you are already talking about it. And with corn oil, you have the first generation of it. With that, I thank you very much for your attention and hope you were able to take something away from the presentation.
Thank you. We're open for questions. Oh, there. Mike.
Hello, Dr. Wen. Good evening. Please tell me one thing. There is a confusion when we process maize, whether jet cooking is essential or it can be done without jet cooking, liquefaction.
Yes, I would say that in the US, we see more jet cooking than not jet cooking, and it can aid in solubilization and breaking up the structure.
No, you mean out of the both, what is good for maize?
Of the both?
Yes.
Of the both, what?
Jet cooking at 108 or so, or a 190-95 degree liquefaction.
For release, jet cooking is better.
For release, yeah. Because there had been a debate going on whether jet cooking was done outdated in our industry in India. And everybody was going for low temperature liquefaction. Even the enzyme people were also supplying that, no, you go for that. But that is not a good idea, in fact. It should always be jet cooking first, so that pop-up is there. So you do most of them with jet cooking?
Yes, yeah.
Thank you.
Yeah, even in the Indian plants, we have seen that the shear generally helps. But having said that, a lot of our customers use mix, right? So when you have a lot of rice there, then I would say that you can optimize your method, like the liquefaction, to even bypass it. And of course, there's some amount of enzyme optimization required. But for 100% corn, their jet cooking definitely helps.
And when we mix rice and maize and process together, then low temperature liquefaction is okay?
Again, depends on the mix, right? So if it is more heavy towards rice, then yeah, slightly lower temperature can help. But then if it is more towards the corn, then I would say higher temperatures. And we have now enzymes that can actually do wonders at the higher temperature. Because earlier there was always this, stability issue. But yeah, Ben just talked about the phytases. They really help improve the stability of the enzyme. So you can go to 95 and without really impacting the enzyme.
No, tell me one more thing. If I have a rice mix more than maize, then the jet cooking will help or not?
Then you can bypass jet cooking, yeah. Because rice is an easier starch.
It doesn't help.
It doesn't help. Yeah.
Okay,
yeah.
That's right.
Yeah.
Thank you.
I have two questions. So about 25 years ago, India did not manufacture cars. Okay. The first time Maruti came and then we started manufacturing cars. Today we are the largest supplier of auxiliaries of car all over the world. In the case of enzymes, the complete Indian industry is dependent on imports. Companies like yours, are you not thinking of setting up? Everything is today make in India, make in India. We are manufacturing, even talking of F-16 manufactured in India. Why the enzymes we are not able to manufacture? This is one thing. And the second question is, in the introduction of yours, it was mentioned biobutanol. So please teach us how to manufacture biobutanol, because about ethanol, everybody is expert here. So if you can bring something like this, I think it will be a good service to the society and to this country. I shall be grateful if you can share your thoughts on these 2 points.
Sure, maybe I'll take the manufacturing question first. Yeah. So, as you all saw that the ethanol industry has just recently started to, really expand so much that the entire volumes can be taken, because these, manufacturing in India means you will generate a lot of enzyme. And of course, always the question was, can we even consume here? Now that's not a worry. So we are looking at one of that option, and hopefully soon come up.
Madam, actually enzyme is not only used in ethanol. Ethanol manufacturing is only a fraction of it.
Yes.
But real amount of enzymes used in across the board in different industries. There are fellows sitting here, pick up 1 or 2 from this. Maybe our company or maybe any other company which you like. Let us start manufacturing here in India and teach us what should we do so that India becomes the manufacturer of enzymes. Yeah. And you earn from your technology whatever fee you have, and please make enzymes manufacturing in India.
Okay, biobutanol. So there are actually a couple people on the stage who spent part of their career on biobutanol, biobutanol development, which was the fermentation. So basically going from glucose directly to biobutanol, no intermediates or so. I spend a lot of my time at the demonstration facility for this process, and actually we have commercialized part of this process for a short period of time in the U.S. But ultimately, the biotech process from glucose to biobutanol has not been large-scale commercialized, and I think. Part of the challenge, obviously, we're talking about how high can you go with ethanol concentration in a fermentation, right? I think you have to take a factor of 8 or more to this number when you think about the tolerance the yeast has to butanol. And what that really means is that you have to solve biotechnology through process. And it is always challenging to solve biotechnology through process, because what it means is you carry a lot of water would be one example, right? So, so the technology as originally maybe intended has not been commercialized in the US.
Can ethanol be mixed with diesel?
I would have my doubts.
You have doubts. There's the octanol, what you are saying, butanol can be mixed with diesel?
I'm not sure that it can be mixed with diesel, but it certainly, from a US perspective, has properties which definitely changed the infrastructure or didn't require the same infrastructure modifications potentially. So,
okay, thanks.
Yeah, you could get to a higher blending rate with butanol for sure, much more than ethanol. Yeah.
Madam, butanol is actually non-aqueous, it is non-polar. Yeah. You can blend any amount in diesel. Yeah. Ethanol you can blend in diesel. Butanol you can blend in diesel. Ethanol, no. Yeah. Isopropanol slightly more than ethanol. Ethanol being highly polar, it's not possible in diesel. In petrol, it goes well because the polarity matches. To blend ethanol in diesel, you are supposed to add emulsifier. But then along with water and ethanol together, you can do.
Hi, are you guys working in India to get GM yeast, or are you guys already have started GM yeast in India?
So, can you hear?
Yeah.
So, we are one of the few companies who's already got an approval for the import and marketing of GM yeast. And, as a regulatory requirement, any ethanol producer who wants to use GM yeast actually has to also go in through a regulatory approval process. And we are already working with some of our customers to put in the application and, get the approvals so that, they can start using the GM yeast, which brings in a lot of, benefits to the ethanol producers. So we're already on it, yeah. Hopefully next week we start some trials of GM yeast at one of our customers' places.
Actually, I have one question. This side. I have one question. Can you tell us what are the advantages when we use GM yeast vis-a-vis the normal yeast?
So yeah, maybe I can take that. What we did in US already we had seen a lot of advantages, but because it was important to also know under Indian conditions, what those advantages can translate into. So we have seen that depending on the process conditions, let's say you are, exposing it to very harsh conditions, then you see almost a 3 to 4% increase in yield. There's a significant byproduct reduction, right? And then the other is the ease of operation, right? So right now we struggle with controlling the temperatures, and especially in the seasonal, hot season. The GM yeast are much more robust. So they can, really make your operations easy. So you don't have to worry about that there would be a stuck fermentation and, everything kind of, that with the normal yeast that we sometimes see, it temperature goes above 38, slows down, maybe even leaves sugar. So all of that, has been, I would say, engineered into the genome. So these engineered yeast can do a much better job there.
So you are saying it's only ease of operations, or we get better ethanol yield also?
That's what I said. The first is 3 to 4% better ethanol yield with byproduct reduction, which is glycerol, and also it takes, controls contamination. These are very fast. So what it does is in the first 16 to 17 hours, you'll see suddenly a lot of ethanol produced. So your contamination is better controlled. So all that yield improvement comes from that.
Okay, so you are saying if we use the yeast which you are providing today with the GM yeast, we will see a
3 to 4, yeah.
And in addition to that, you'll also have a reduction in the glucoamylase consumption. So right now you use two, enzymes. One is the alpha amylase, which goes into the liquefaction area, and second is the glucoamylase, which goes into the fermentation area. So, when you use a GM yeast, depending on, the type of yeast, the GM,
how much is the
that have been
reduction?
made.
How much is the reduction of glucoamylase dosage?
At least our yeast, we, talk about a minimum of 50% reduction in the glucoamylase.
Yeah, it can be more. It's at least a 50% reduction.
Yeah.
Like
Okay.
across
I'll talk offline.
Okay.
Sure.
We have another 3. Hello.
Yeah, hi Ben. My question is whether the GM yeast will only work to increase the yield, or we have to put some chiller also in fermentation, because as India, it's a tropical country and we have high temperatures during summers, and most of the months are really hot. So where temperatures are going up to 50 degrees Celsius, and maybe in future we can see the higher temperatures as well. So whether the GM yeast will only be working just to get the higher yield, or we have to think about temperature control in fermentation also?
I'll take. So I'll say the GM yeast are much better than the non-GM yeast in terms of temperature robustness. So if your current, non-GM yeast, you see some struggle, 35 degree plus, right? So these yeast can take that, temperatures much better, up to 38 we have seen, they continue to provide additional yield. So yes, of course, you get the additional yield, but also I will not claim that you can go to 50 degree, right? So there is a limit to, because these are still living organisms. So, at some stage they will struggle, but yeah, they can definitely do much better job than the conventional yeast. And of course, we continue to innovate, so who knows, yeah.
Okay, so 38 degrees Celsius they can tolerate.
Absolutely.
Okay.
Yeah.
Above that, we have to think about temperature control and all other things.
And the other thing is, it's not just the temperature, like one temperature, it's also the fluctuation. These yeast can better, fare these fluctuations. And that is what we often, the yeast struggle with, like, the sudden fluctuations going up and going down. So those, so you can.
I think this will be the last question.
Thank you very much. What about the cost difference between the GM and non-GM yeast? Approximate?
So at this point in time, we have not come up with a commercial number for the GM yeast. We want to first demonstrate the efficacy of our GM yeast. And I'm sure we will be offering our customers something that they will have a takeaway. I'm sure unless.
Can we pass the mic here quickly?
You are saying that the registration process is on and everything. So can you just explain a little bit about how it's getting approved and when is it likely to get done?
For the user, so as Arun mentioned, right? So we already have our yeast approved. Now, the user, which is the ethanol producer, also needs to put in an application to a regulatory body called GEAC, right? And what GEAC says is, before you do that, you establish a institutional biosafety committee at your plant. Now, this is your committee, but there is a nominee from DBT, which will sit in that committee. And now this committee will do the oversight of the entire process. Now, somewhere between, I would say, 3 to 6 months is what this takes, because you'll have to establish the IBSC, get the yeast approved, whichever yeast, whether it is our yeast or other yeast approved, and then go to GAC. So somewhere between 3 to 6 months. And right now, as Arun again mentioned, we are supporting our customers in putting in their applications here.
So is any factory approved in India yet?
Sorry?
Is any of the factories approved in India yet?
Yes, we've already had a couple of approvals, yes.
Excuse me, ma'am.
I think we will have to conclude this session now.
This is the last question, ma'am, please.
Where I can't see.
Yeah, this is the last in, ma'am.
Okay, very quickly, please.
Actually, ma'am, you just claimed 3 to 4% of increase in yield. Where are you thinking this recovery will come out? Because right now you are assuming this is come from any byproduct.
Yeah, so again, the 3 to 4% recovery improvement partially comes from the byproduct reduction, right? It also comes from the fact that the yeast are able to tolerate. So, the contamination and fight the contamination better. So, because a lot of sugar or, carbon that we lose to contamination as well, right, today. Because if the yeast is slow, the contamination picks up. We see oftentimes 0.4 to 1% lactic acid, right? So, if the yeast grows better and is able to make ethanol better and your contamination goes down, so all of that carbon also gets diverted to ethanol. So we called it yield and use, improvement. So that is 3 to 4%. So it is a combination of everything.
No, ma'am, just need to know whether we have a data.
Yes, of course. I'll connect with you offline and we can show you some data on that.
Okay.
Yes, certainly.
Or during tea. All right, thank you so much, team. Let's please give them a round of applause for their presentation. Thank you so much, Dr. Ben. And could you also please introduce your team to us? Could you introduce your team to us? Could you? You did, right? Okay, great. Fantastic. I missed it then. But thank you so much. It was wonderful having you guys on the stage and for a great session. Please have a seat. Thank you. After a photograph.